So polymerase chain reaction and its application. So first, let me define what is polymerase chain reaction. So polymerase chain reaction is, I would say, the same thing like replication. The only difference is that you are doing or you are actually replicating your DNA. You are making your DNA inside one tube. That is happening outside the cell. So that is polymerase chain reaction. And what happens here? The PCR is an exponentially progressing synthesis of the defined target DNA sequence in vitro. Let me explain that. So here, if you have one big long DNA, something like one human DNA, like long DNA here, and it can be too long and so many sequences there. Maybe you are interested only on this portion, like this, you are interested on this portion of DNA. And now you want to make more and more these sequences or this portion of the DNA. So what you will do, you can do this PCR reaction or polymerase chain reaction where you can amplify this thing. So for example, initially you have just one DNA or just two DNA molecules. Then you will see after 2 hours, 3 hours, you will have million of this DNA. I will explain how you can get this thing. But this is very important because initially I introduced that this PCR is very important in normal academic research. Like some one of us, we are interested on some protein because of their therapeutic purpose. Like some protein are being used in therapeutics, as therapeutics, I would say. And also, uh, as I told you before, that in forensic lab, when people get some kind of biological sample, some fluid, some hair, some other things, if they want to establish like who is the victim and who is like all those information you will get after doing thorough forensic test, and DNA sequencing is one of them. Like there are many other forensic tests, but DNA sequencing is one of them.
So, one question. So, can you
Yeah,
just please explain which part we amplify here? Like, I did not understand.
yeah. I will. So the detailed mechanism I will come into that right now. Okay, Tanmay. So this is just introduction. I will discuss in detail. So now the we need to sequence the DNA, but how you will sequence the DNA? Because you need certain amount of DNA in order to sequence it. So before that, we need to amplify the DNA. So now what Tanmay asked, that what portion we will amplify. So initially I would say that if we are concerning about some crime related to some human individual, and now we have the whole genome sequence of human, so we know what is the sequence of human, and the sequence difference between me and someone of you, Tanmoy asked, I would say the difference is very little because both are human. So there are some kind of primers, like I will come into that again, in more details. But our goal is to just amplify DNA. At this position, you don't need to consider that which portion of the DNA we want to amplify. After explaining the mechanism, you will get it. And then whenever you have like amplified DNA, then you have to sequence it. Then if you sequence it like a long stretch, then you will find that there is some kind of differences between my DNA and someone of your DNA. Because although both of us are human individual, that's why most of the sequences are same, but still you will get some minor differences. From that differences, you can establish the person who is involved in that crime or whatever, like sometime for identity issues and all other stuff, you have to sequence DNA. Even for some treatment, some medical condition, for that reason also you need to sequence DNA. So now come to the mechanism. So PCR is an exponentially progressing synthesis of defined target DNA sequences in vitro. And now the question is why it is called polymerase chain reaction. I will come into that. And before that, I want to tell you that in 1983, this is not very old. Like, if you see most of your, like whatever you studied in physics, chemistry, most of them, they are like, they have been discovered long time back. But relatively, this is very new. So in 1983, Carey Mullis actually invented this procedure, and he got Nobel Prize in 1993 because of its huge application in biomedical field. Now, the first question here is why it is called polymerase chain reaction. Why it is polymerase? Because I am telling you that this is similar to replication, but it is happening in vitro. It is called polymerase because, see, in case of DNA replication, I know that most of you actually face a lot of problem there. Many enzymes are involved there, like at the beginning of this class today, someone asked already. So helicase, topoisomerase, primase, DNA polymerase, RNase H, lot of enzymes there involved. But although we are doing the same thing in vitro means not inside the cell or not inside the body. It is happening inside tube in lab. That is called in vitro. And inside the cell, inside the body, that is called in vivo. So we are doing the same thing, but inside the tube. But here we need just one enzyme. Nothing we need. Just polymerase is good enough to do all the function, right? So here that is the very important thing here. So that's why this is called polymerase chain reaction, because just one enzyme is sufficient. And this enzyme specifically is called Taq polymerase. I will explain what is Taq polymerase and why it is called Taq polymerase. And the next point is why chain. It is called chain because the products of the first reaction become the substrates of the following one, and so on. So that's why this is just like a chain reaction. Now, if I come into, or if I ask you actually, if we need to carry out a PCR reaction, right? Then what will be the ingredient? That what ingredients we actually we require? Can you tell me? Let someone of you just unmute yourself and tell me what are the reagents required in order to carry out polymerase chain reaction.
Sir, enzymes, template strand, and this DNTP is for the nucleotides, means making the chain.
Yeah, so that means all four nucleotides, template DNA, and enzyme. You are right. Only one thing is missing here. Important thing. Someone can remember that what is missing here?
Mg2 plus
DNA polymerase. DNA polymerase.
Uh, no, so the polymers he already mentioned that enzyme. So
Sir, we
Mg2
have to
plus, sir.
first
Mg2 plus.
extract that template strand from some method.
yeah, template also, like template DNA also he mentioned the primer, right?
Mg2 plus, sir.
Yeah, magnesium is required. Yes, so two things are missing here. Magnesium ion, that is required because I told any DNA polymerase for their function they require magnesium, and also you need primer. Okay, so here primase or other enzymes are not involved because we are doing it inside tube. So we will add primer actually from outside, and that will address someone question on my question here, that which portion we want to amplify. I will explain that. So let me first show you what are the ingredients. Already I asked you. So polymerase chain reaction, you need the reaction component here, like target DNA, like the DNA which we want to amplify, right? We want to make more and more DNA. And then pairs of primer, that oligonucleotide, that means short stretch of nucleotide that define the sequence to be amplified. This is the problematic part. I believe that some of you might have problem. I am explaining this thing. So here. So if this is the DNA sequence, 5 prime, 3 prime, 3 prime, 5 prime. So here you have like sequence like A, T, G, C, G, C, and you have like, n number of sequence here, like lot of sequence here. Then again, I'm just writing T, C, A, G, C, C, something like this. And here it's complementary strand. So you will have T, A, C, G, G, C, G, N, here A, then G, T, C, G, G, and you can write so many sequences here, whatever present in this DNA. Now, in PCR, what we do, actually, the interesting thing here is you have to use two primers. Primer, the concept of primer you have learned in the previous class. So primer means here one short stretch of nucleotide. So for example, here, if you want to put one primer here, I would say primer sequence will be here A, T, G, C, C, G, C. So this is one primer, right? And this is 3 prime, so this is here 5 prime, and it can be extended in this direction in presence of polymerase and suitable condition. Another primer you can use here, right? So again, the sequence will be G, G, C, T, G, A. This is 5 prime, so it can be extended this direction in presence of suitable condition. So this short nucleotide sequence, like oligonucleotide, sometimes we call oligonucleotide because it should be like around 10 to 12 or 18, 20, something like that, like small fragment of DNA. And here you can use those DNA fragment as a primer. Remember one thing, in case of replication, which is what is happening in our body, in that case we use RNA primer because DNA polymerase cannot start the reaction, but primase can do. But in this case, we are carrying out the reaction inside the tube. We are doing it in laboratory. So we are actually adding this primer as synthesized one. Like we synthesize this DNA fragment in our lab, or you can commercially buy. You have to provide the sequence and they will give you the primer, like in terms of DNA. And then you have to mix this DNA in the reaction mixer. And as a result of that, those primer will anneal in specific position, right? Because of this sequence. So this is hydrogen bonding. And now in presence of enzyme, it will be extended. You have to have nucleotides, magnesium, all those things should be there. So as a result of that, now if you do this reaction, which portion of the DNA should be amplified? This portion, right? This portion, not this portion. So this is not going to amplify it because we are using primer here and here. Why? Because in the first cycle of the reaction, this primer will be extended like this, and this primer will be extended like this. And then again, we will do the next cycle. I will discuss how this next cycle will come. So I know that you have several questions, but I will let me go through one or two more slides, then you will get the idea. So target DNA and pair of primers, and then DNT bits, that means A, T, G, and C. Thermostable DNA polymerase and magnesium ion. Here the most important thing here in this case PCR, you cannot do PCR without this special enzyme. This enzyme we are calling thermostable DNA polymerase. Do you know why this thermostable DNA polymerase is required?
So because might be in lab we require a bit of temperature, like there might be a bit of temperature problems, which is not in the case of body cells because there the temperature is maintained.
Yeah, your answer is towards right direction. So see, intentionally I didn't tell that before. Now I'm telling you. So you have double-stranded DNA as template, and inside your body you have lot of enzymes to overcome the problem like hydrogen bond. You have to break the hydrogen bond, all those things. But inside the tube, when you are doing this, what you are doing? You have this tube and you have like reaction sample, like template, DNA, primer, everything. Just minimize this tube, like make a small tube. And then what happened? You have double-stranded DNA. So the very simple thing, you have to increase the temperature of the solution, the reaction mixture. The first class I discussed that if you hit the DNA, double-stranded DNA, what will happen? The two strands will be separated because the hydrogen bonds will be broken. So now two strands are separated. And now what will happen? Like at high temperature, two strands are separated. Now you are again slowly cooling down the temperature of this tube. Then those primer that will anneal in a specific position where it will match with its sequence. And then those enzyme which is present there, the polymerase enzyme, that will actually continue the reaction, the polymerase reaction. As a result of that, the new strand will be synthesized. Then from one strand, like one DNA molecule, you have now two DNA molecule. In the next cycle, what will happen? Again, you have to denature your DNA. Denature means again you have to separate both the strands. How you will do that? Again, you have to increase the temperature in order to like separate double-stranded DNA. So that's why in every cycle you have to increase the temperature a lot, almost 94 to 95 degree centigrade. And again you have to cool down. Again you have to increase the temperature. It is going like that. Normally in lab we continue up to 30 to 35 cycle. So that if you start with that one DNA molecule, so it will be like huge amount. I will show you the calculation. But inside our body, the temperature is ambient temperature, our body temperature. But in this case, we need some enzyme which can withstand. That enzyme can be functional even at high temperature. After so many cycles of high temperature, but still that enzyme will be functional. That is very important thing here. So that's why we use this enzyme. This is called Taq polymerase. Why Taq polymerase? This is just a DNA polymerase, but this Taq name came from T stands for thermos and AQ stand for aquaticus. If you forget the name, nothing will happen. Okay, I'm just explaining so that you can remember if you want. So thermos aquaticus, T and AQ together Taq polymerase. What's the significance? Thermus aquaticus, this enzyme or polymerase actually extracted or it came from some bacteria. That bacteria generally or normally stay in hot spring, almost like boiling temperature water. You know that life is existing almost everywhere in the earth, even in hot spring, right? Like very hot temperature. There also you will find lot of bacteria surviving oil. That means they have all those kind of properties so that they can be okay in high temperature. So their polymerase is functional even at very high temperature, like 90-degree temperature, right? So as a result of that, like humans, so we are like clever at this step. So we have extracted that enzyme and we use that in our own purpose in polymerase chain reaction so that multiple cycle of temperature increase and decrease, still the enzyme will be okay. It will not be destroyed. So as a result of that, we use Taq polymerase for polymerase chain reaction. And now,
Excuse me, sir.
yeah,
Sir, when we cool the means when we cool down the temp means when we reduce the temperature, sir, how does the original H bonds do not form? Means
yeah, yeah, yeah. That's a good question. That is a kind of very good question. Let me just give 2 minutes because I didn't show that slide yet. Then Rajas, I will answer your question. Yeah, so very nice question actually. A solution buffer is required for maintaining pH as well as ionic strength and those things. And I just finished about the enzyme part actually. And now this is I would say the final slide that where you will get almost everything. So the region of double-stranded DNA to be amplified here. So we are just showing here that 5' to 3' and 3' to 5' double-stranded DNA here. So as a result of that, the first step, heat to separate strands. Okay, so normally in lab, like, this is again, I'm telling you, this is our routine work. So like my research scholar, normally they do almost every week, they are doing this thing, like, like for different purpose. So normally we heat it 94 degrees centigrade. Okay, now those two strands are separated. Remember, in this tube, you have this DNA, you have polymerase, you have DNTPs, primer, everything. So now the next step is cool.
Sir, one question.
Yeah.
Sir, we actually stop heating. Like we have to once it here the strands are separated, we have to cool it down also. So exactly when do we know that these strands have separated? Like it could have been, it could be 50% denaturation also. Like, is there a fixed time
Yeah,
for
yeah,
that?
Tanmay, so normally the 94 degree is too high, okay? So like when we discuss about TM, like melting temperature of DNA curve there, so you will see 94, 95 is too high. Like any DNA will be melted. Melt means like those two strands will be separated within few seconds, 30 seconds. Normally that cycle that when we are increasing the temperature, when it will reach at 94 degree, then we keep there around 45 seconds. Right. So I will show you that cycle that and this is good question, but just let me go through this slide. I think you will get this answer also. But in order to provide your first answer that 50% denature, so I would say at 94, 95 degrees temperature, like full strand will be separated. If you are doing like 78, 80, 86, that is like, full population or the all population will not be separated. Okay.
Yes, sir. Got it.
Yeah. So let me, and then Rajesh actually asked one more question. I'm coming here now. So now, when you are cooling down the temperature of that tube, then what will happen? Two things might happen actually that he pointed out. One thing is that this parent strand, right? Whatever you have these two DNA strand, that can also again, like, anneal means they can make again double stranded DNA when we are cooling. And the other thing what we want is we want to anneal this primer. Anneal means this formation of double strand again. So that primer are actually attaching with the template because of their sequence. Now I would say the number of primers are very, very high actually. This is some kind of competition, I would say. Like, so primers are huge in number, in like I would say like 1:1 to 1,000 or even more. So as a result of that, like I would say some of those DNA they might like anneal together, but some of them will be annealed with the primer itself. Because here we are showing just one double-stranded DNA at the starting point, but actually at the beginning you might have like 50, 100, something like that. So as a result of that, you will get this kind of scenario, and also you have huge amount of primer. So you are actually ignoring the double strand formation between the original or parent strand. And now, whenever this primer will be annealed, then the third step is already you have DNA polymerase and nucleotides are there. So then the enzyme will actually elongate. Elongate means synthesize the DNA. See, you started with one DNA molecule here, just for explanation, double-stranded DNA. Now what you have after all these steps, you have two DNA molecule, right? And this is just one cycle. And the next cycle, what will happen? Again, in the next cycle, you will hit the, like, you will increase the temperature of the tube. So as a result of that, you can see it will be again separated those two strands, and also the another DNA molecule like this one, it will be separated. So then if you do all those reaction, like all those steps, you will get 4 DNA molecule. From 4, again you will get 8. So it will be continued like this. That's why I told you that it will grow in exponential manner. So finally you will get lot of DNA molecule just after 2 to 3 hours. And let's see how the reaction cycle looks like. Something like this. This is not like, exact, because this is depending on many things. So at this level, you don't need to understand, because how big your DNA molecule, like how much you want to amplify, like how much you want to sequence, and then what's the kind of sequence of the primer. There are a lot of things you need to take care of it. But it looks like the overall, it looks like this kind of thing. This is the cycle, like as I told you, 94-degree, 95-degree. This figure I have taken somewhere where they use 98 degrees centigrade. And then this is the annealing temperature. That means you are actually cooling down so that your primer can be again bind to the template DNA. And then you are increasing the temperature a little bit so that the enzyme can work better. Enzyme means Taq polymerase. The thing is, Taq polymerase, that enzyme is very nicely work in high temperature. So that's why we are increasing the temperature again. So now